Hello, Professor. My name is Nidia Apostle. This is the lab number 9. What are we doing here after we clean our surface with 10% solution of bleach and gather all our supplies? This is going to be the video experiment of the environmental influence microbial growth salt tolerance. So what we have here is our previous growth for S. cerevisiae and S. epidermidis. Then we have different concentration of salt. We have 1%, 7%, and 15%. And what we're doing is we're transferring 2 drops of the growth of the S. cerevisiae and epidermidis to the tubes for the salt, so we can see the different osmolarities that can grow. We're going to incubate for 48 hours. After the 48 hours, we're going to check and be sure that bacteria has growth. If there is no growth, we can add another 24 hours. After that 24 hours, if nothing grows, there will be negative test. So I have my pipettes, I have my tea candle, so I can do the aseptic technique. So we're gonna begin. So as always, we're gonna grab the tubes. This is my growth that I have from previous experiment, and this is the 1%. Aseptic technique is going to be done in both of them, and once we do the both, we're going to take our pipette that has been seated in alcohol. Be sure that it's dry. We're going to take only 2 drops from there, and we're going to pass it to our 1% solution of salt. We're going to go ahead and do the aseptic technique. We're going to cap it. This is done. Then we're gonna go ahead and sterilize this one and continue with the 7%. We're gonna go ahead and sterilize also the 7% to be sure that we're doing the aseptic techniques. So again, gonna take 2 drops, and then we're gonna move the 2 drops to there, put it back, sterilize again so we can cap it. So we're gonna go ahead and cap it, and we're gonna put it away. We're gonna sterilize. The aseptic technique is so important. Also keep it away from your nose to not aspirate or breathe into the growth bacteria. We're gonna sterilize appropriate both of them. We're gonna grab the pipette in the alcohol, be sure it's dry. Again, we're taking 2 drops, moving the 2 drops and back. We're gonna sterilize, antiseptic technique, and then cap it. So we have the 1%, 7%, and 1, 7, and 15 for the cerevisiae. We're gonna cap this, and we're gonna keep it for our next experiments. So we finished with that. We're gonna move now with the S. epidermidis, and we're gonna go ahead and do the same process. So we have the 1%, 5%, 7%, and 15%. So 1, 7, and 15, different salt concentration. We're gonna go ahead and take our growth that we have from previous experimentation. We're gonna go ahead and do the aseptic technique. That's done. Now we're gonna do the 1%, and we're gonna do the same. This one is gonna be moved to the trash because we finished with the cerevisiae, and now we're gonna move to the next pipette, which is for our epidermidis. So when both are sterilized, we're gonna do the same. We're gonna go ahead and take only a little bit to do the 2 drops. Pass in the 2 drops, put it back in there, sterilization or antiseptic technique, and there it is. So we're gonna go ahead and cap our experiment and put it here. This one continue being sterilized. We'll continue using it, and we're gonna sterilize the 7% salt solution. This is the salt tolerance experiment. Remember, be sure that it's no alcohol. We're gonna grab some drops of the previous, put the 2 drops in there. We're gonna go ahead and do the antiseptic technique. We're gonna go ahead and cap it. We're gonna move it to our metal. We're gonna continue with the sterilization. I'm gonna move this so the cameraman can catch that very well. Continue with the sterilization. Antiseptic technique also for our empty one. So here it is. Same process. We need to be sure that it's dry, nothing in there. We wanna catch the 2 drops, pass it to the tube. Again, antiseptic technique. Antiseptic technique. We're gonna go ahead and cap it. Move our experiment here and finish antiseptic technique for our broth that we already have cultured for the epidermitis, and we're gonna keep it in the refrigerator for the next experiments. So after we have finished, we put this in the bleach solution. We have 6 of our broth now with our experiment. This is going to be an incubation for 48 hours until the next video.